So welcome everybody to this session. We are going to have 4 different talks about different topics related to biological systems. Our first speaker is going to be Shweta Bansal. She comes from Georgetown University, and she's going to talk about social behavior, network structure, and wildlife disease transmission. And before you start, Shweta, I just would like to tell the participants that you are welcome to ask any questions in the chat, and at the end of the talk, I'll read them, and Shweta will answer for as many questions as we have time. So, well, going back to the talk, please, Shweta, whenever you're ready, you can start.
Great, wonderful. Thank you so much, Marta. Hi, everyone. I want to thank Panos and the entire CCS 2020 team for their work in putting together this meeting in this unprecedented year, and also, of course, for the kind invitation. My group has been thinking a lot, of course, about COVID research for the last 9 months, as so many of us have. But today, I just wanted to take a break from some of that and talk about our broader work on social systems through the lens still of disease transmission. So, sorry. So complex social systems are, of course, composed of the behaviors of individuals, and as all of you will well know, these are characterized by interconnectedness, nonlinearity. They're adapted, and when you think of complex social systems, you might think of one of the many social systems and structures that underlie human societies, such as schools and workplaces and hospitals. But social living is, of course, actually a much broader phenomenon and is found in many animal species that live together, develop social bonds, and cooperate towards common goals. And the benefits of social living result in more efficient foraging, mating, higher chances of surviving. But of course, the big cost of living with others, as we're all learning this year, is infectious disease risk. And the traditional view when it comes to sociality and its role in disease transmission is that sociality increases group sizes, which in turn increases transmission. But the evidence over the last 20 years has really shown us that things are more complex than this, and that group size just isn't sufficient for thinking about and being able to predict where infection prevalence will be high. So this has really highlighted the need for sort of a complex systems approach that I think this audience will appreciate, where instead of just taking into account one feature of a social group, we holistically consider its structure. And of course, there's been a lot of development in the idea of modeling disease transmission as networks in humans, but with respect to wildlife diseases as well. This sort of paradigm has really taken off both empirically and theoretically. And there have been some really important success stories that have occurred in the last 10 to 20 years. But the field is still growing, and there's still a number of gaps, which are really gaps in the study of all complex social disease systems. So conceptually, we still need a better understanding of the relationship between individual and population level sociality. As when it comes to disease risk, we need to understand how social systems mitigate disease risk and how social systems might actually drive the evolution of the pathogen, of the infectious agents that are spreading over them. Empirically as well, there's a lot of work to be done to better integrate empirical data with theory and to also be able to compare across systems. So, of course, so many of these, both conceptual and empirical questions can be done and can be addressed in humans, but really to be able to look at this beyond one single type of social systems that humans represent, we really do need a broader perspective. So for that, my group's developed ASNR, which is an animal social network repository that, thanks to the sharing of data that has really become popular over the last decade or so. This has allowed us to put together a big repository of networks from over 70 species including mammals and birds and fish and reptiles and insects. And the repository at the moment includes 700 networks from these 70 species, and we're really hoping to continue to grow this over time. So being able to use data from across taxa can really give us a broad perspective as to the role of social systems in disease transmission, and how we can actually better understand social systems through the lens of disease transmission. So today, what I'd like to do is give you a sample of the work we've been doing to address these gaps that I identified using this large data repository. I'll go quickly through 3 research questions that we've looked at recently, and I'd be happy to, of course, talk with anyone afterwards about the details of these studies. And all the work that I'm presenting today has been led by 2 stellar young scientists. Melissa is a current PhD student in my group, and Bertocchi was a former PhD student with me and now is at Yale University. So to get started with sort of the first case study I'd like to talk about, I want to talk about how social systems can come in many shapes and sizes. So of course, as humans, we represent one kind of social system, but non-human species show a diversity in their sociality. So at one extreme, you might have fairly solitary species and populations in which adults really only interact during their breeding period, and there's not a lot of other cooperation and social bonding going on besides between offspring and parent. And somewhere in the middle, you might have species that are more gregarious, right, where they aggregate to do one or more activities. But this aggregation isn't particularly stable in space or over time. And so the group composition is constantly changing. Fission-fusion species like bison or like dolphins or like humans fall in this category, as do many bird species, which tend to be quite gregarious, but not in a stable way. Lastly, we might have socially hierarchical species, which have this very strict social structure that is permanent and is long-term, and it's usually structured in a hierarchical way. So many insect species, carpenter ants, for example, fall in this category. So as a first question, we wanted to understand, can network structure itself quantify this type of social system structure? These social systems have been classified by ecologists that are experts in these various, in each of these species, that study them over a lifetime and observe their behavior, and that's where these classifications come from. But our first question was, is this sociality actually reflected in social network structure? So for our approach, we took the data from across taxa from this social network repository, ASNR, and we analyzed them through a Bayesian mixed model framework, so a regression framework, in which the response variable was sociality, and the predictors were various network metrics, both individual level, local scale, and global scale. And then we controlled for a few different things in terms of the data collection process and what the relationship between the different species was phylogenetically, and so forth. And just to talk about some of the results, we found some really interesting and in some cases surprising things. So one, we found that relatively solitary species actually have a high degree of heterogeneity. This was not my intuition going into it. I would have thought the more social species in the data set, species like humans would have more variation in how many individuals they contact. But it turns out actually the solitary species have a lot more heterogeneity in their contact. On the other hand, gregarious species tend to be more fragmented, and this is something I'll come back to in a minute. And then the socially hierarchical species tend to have a lot smaller clustering, so they have much fewer triangles in their networks. What was particularly surprising and sort of the big caveat to these results is that these differences were actually primarily driven by low weight edges. So if you took away the edges, the contacts that individuals had that were of low duration or frequency, then the differences between these social systems actually went away. So this is something where hoping to dig more into, and I'd love to chat with anyone about, is that really it's those sort of casual contact that we have as humans or that wildlife have that really dictate the true structure of social systems. Next, we wanted to ask whether, sort of what the role of sociality is in disease transmission. Of course, we, all have many of us have a very good understanding, and the field has a very good understanding of how network structure is connected to disease transmission, but how does sociality dictate disease transmission? So again, we used data from the social network repository. We essentially modeled infectious disease transmission using a simple SIR model with varying parameters on each of these empirical networks. And then again, we used a regression framework where in this case, the response was a disease consequence, either epidemic size or duration or probability. And the key predictor was sociality. Of course, we controlled for a few other things. And so the results here basically summarize 4 different types of social systems, depending on the pathogen transmissibility, what the epidemic consequences, in particular probability and duration are. There's a lot going on in this figure, so I'll try and summarize. So basically what we find is for high transmissibility pathogen, for a very contagious pathogen, we find that there's sort of frequent and prolonged outbreaks in gregarious species, in these species that are fairly social and are constantly changing their contacts. Whereas for a low transmissibility pathogen, for a low R0 pathogen, for example, the outbreaks tend to be a lot more prolonged, especially in socially hierarchical species. And this reflects the network structure findings that we found on the previous slide. So sort of to summarize what we've learned, this was certainly one of the findings that was surprising to us is that only really a few network features. Again, as a reminder, we put in about 12 network features into this work. We found that only a few really mattered in distinguishing social systems. And really, even those differences were really driven by weak ties, by these low edge rate connections between individuals. And we do find some evidence for social structures providing protection against at least a limited set of pathogens. And so really looking at how network structures and different social structures have evolved alongside pathogens to protect the populations that they spread in is going to be an important step. So that brings me to sort of my second case study, where we really want to investigate particular structures that can help mitigate disease risk. So why we expect such structures to exist is because social living, as I've mentioned before, comes with trade-offs. There's a benefit to social living. That means better access to resources and better access to protection from predators. But of course, the cost of social living is that you can easily be infected with an infectious disease, and that certainly reduces your overall fitness. So one hypothesis is that we had was that social systems perhaps evolved to have modular structure or community structure, which might balance these trade-offs of social living. And so we decided to, again, look at this repository of social network data. At the time, we only had about 40 species in there. And so we looked at that range of species, and we looked at the modularity levels in those species. So this measure on the x-axis here is a modified version of Newman's modularity, the Q measure, and it's been modified to be comparable across different networks of different densities. And so with this histogram, we see that at least with this sample of about 45 species, most species tend to have low to moderate modularity, and we don't really find too many that are in that range of 0.6 and above. Then if we look at sort of just by simulating outbreaks in these networks, we find that really the outbreak sizes in these populations are exactly where we would expect if we didn't take the modular network structure into account, and really only the differences come in for the very modular networks, the networks with modularity, relative modularity of 0.6 or higher. And so this empirical work sort of gave us an idea and gave us some hypotheses, but there's of course a ton going on in these empirical networks. The data have been collected in different ways and measured in different ways. And so we really needed a theoretical approach that essentially allowed us to do systematic, like controlled experiments. So we developed a random graph model that had a tunable level of modularity alongside the ability to control its degree distribution. And the key here was that these networks had these two properties, degree distribution and modularity, without without altering any of their other network structures and network features. And they weren't different from random networks. And so we took a range of these random networks, created networks ranging from relative modularity of 0 to 100%. And we again looked at disease outcomes in this population. And what we're looking at here, the heat map sort of shows epidemic size, with darker colors being larger epidemics, and on the y-axis here, this is varying levels of pathogen contagiousness or R0. And so we find that really for even with increasing levels of modularity, the epidemic consequences in terms of epidemic size, really things stay quite consistent. And it really, you need to get to a very high level of modularity, 0.6 or above, to really see some impact of modular structure on disease dynamics. And if you'll remember from the histogram a few slides ago, there's only very few networks that we find empirically that fall within such high levels of modularity. We also find, so I'm not showing it here, but in the paper, we look at some of the more specific mechanisms that underlie this result. And we find that effectively, so disease has to get trapped in modules for modularity to really have an effect on final disease consequences. Also, I will mention that even though we do, the overall disease burden does not change with modularity, we do find that as modularity increases, the proportion of transmission events that stem from local or within module interactions versus global or between module interactions really trade off in this very smooth way as modularity increases. So to summarize a little bit here, our hypothesis doesn't seem to be supported. We don't find that modular structure in social networks exists to alleviate infectious disease risk. It might exist for other reasons, but it's not necessarily there for alleviating infectious disease risk. But we, do hope with this study that we were able to show that looking at empirical network data across a range of species and sort of using that to identify patterns and generate hypotheses can be quite effective, as long as we have a way to compare across those network systems that have been collected, where the data have been collected in different ways and the network structure varies. We also think that sort of approach becomes quite powerful when it's paired with a theoretical approach in which you can essentially do controlled experiments, systematic experiments, to better understand how function relates to structure. And I've got a link here to the random graph generation generator that I spoke about a few slides ago. Lastly, the last study I'll share with you all is the most recent one that we've done, and here we are hypothesizing whether social interactions can actually drive pathogen traits. So when it comes to social behaviors in the field of network epidemiology or complex disease systems, we're all sort of used to thinking about contact influencing disease spread, and we think of contact as this one monolith thing, right? But really all animal species, humans included, have a variety of different interaction types that are relevant to their social systems functioning. So you might have intimate contact where fluid is exchanged and strong social bonds are required for those interactions to happen. You might have physical contact that just exhibit the social bonds but aren't necessarily of an intimate nature and aren't repeated and persistent over time. You might just have proximity contacts where they just come from sharing space and don't necessarily require any sort of even weak social bonds. And finally, you might just have indirect contacts that again come from shared space use or shared resource use, like shared food sources or shared water sources, where those contacts don't even have to happen synchronously. Where in this example, you might have a bird feeder where some birds feed and at that feeder at one point in time, and a different set of birds comes in along and feeds at a different set of time. But for certain interactions, that would still count. In each of these behavior classes sort of translate to transmission of diseases, but the transmission of different classes of diseases. So the intimate contact might be relevant for sexual transmission of diseases like HIV. The physical contact would be for skin transmission of pathogens like MRSA. Airborne transmission is what's spread by proximity contact. An example might be TB or measles. And indirect contacts are relevant for environmentally transmitted pathogens like norovirus. And so our question here really was to identify, does network structure vary with behavior class? So again, we used data from the ASNR looking at networks across a range of taxa, and we again used a regression framework. In this context, our response was the network structure, and our predictor was behavior class. And again, we controlled for a few things that we knew to vary between these data sets. And we find some results that basically highlight how network structure varies by behavior class. So we find that for skin and airborne contacts, those contacts and those networks have fairly high density. And this comes from the fact that you don't necessarily need social bonds in those contexts to have those interactions. Whereas intimate contacts, have much lower network density, but they have a fairly high degree heterogeneity. Additionally, intimate contacts are fragmented and modular. That's supposed to say not clustered. My apologies for the typo. They're not clustered, but they are fragmented and modular. And so, these results sort of fit with our intuition that the field of disease modeling has, that sexual networks tend to have very heterogeneous degree distributions. Some have suggested that they're scale-free, others find that they're not, but the point is that there's a lot of heterogeneity in those degree distributions. We also believe that respiratory infections, like measles, spread in fairly homogeneous settings, and this is why homogeneous mixing SIR models have been quite effective in modeling those pathogens. So these results certainly fit with our intuition, but it's I think it's useful and necessary to look at this question and not just humans, and to look at it in a range of systems. We also then find that the network structure can affect pathogen fitness. So because these sexual networks tend to be a lot less connected and have more degree heterogeneity, we find that the minimum transmissibility required for emergence in those systems is higher. And so then this begs the question of sort of how do these pathogens, these sexually transmitted pathogens, sort of respond to this requirement of having a higher transmissibility. They can maximize their transmissibility in one of two ways. They can either be very contagious per contact, they can spread more easily, or they can infect the infected individual for a long time, so it can have a high infectious period. And so what we find, we did a review of just pathogens from the literature. These happen to just be human pathogens because that's where the data is best. And we find that actually it seems like the evidence is for sexual pathogens, they tend to have a much higher infectious period rather than higher risk of transmission. And there's certainly theory in disease ecology that tells us why this might be. So it's useful to sort of see evidence for this in this context as well. So just to summarize here quickly, I want to, we're hoping this work highlights that a disease contact network isn't just one thing and is rather just a suite of structures, and really the transmission mode dictates the structure of that network. And I think hopefully with COVID-19, we're going to start thinking about this context a little bit more and really think about how it might in fact affect disease dynamics. And the other thing we're really hoping to highlight here is that network structure, of course, we know affects transmission of diseases, but it might also very much affect pathogen traits. So to just summarize, since I'm almost out of time here, I want to just leave you with two sort of parting thoughts. One is, the data boom in behavioral ecology really has allowed and opened doors for meta-analytic work with network data, and it really is providing an avenue for us to consider some really important and fundamental, not just disease ecology and evolution questions, but general behavioral ecology and evolution questions, and really considering social systems through the lens of disease transmission can really reveal important structures. So I'm hoping that those of you who study social systems but aren't necessarily interested in diseases might still consider them, because that lens can really help us think about social resilience, social complexity, and social evolution at its core. And certainly in a post-COVID era, I think this will be particularly relevant and necessary. So before I go, I want to make sure to highlight again, of course, the great group of junior scientists that lead this work, some wonderful collaborators, both empirical and theoretical, that have been part of our work, and a big shout out to the data sharing partners that without whose generosity of sharing their hard-earned data sets, this work wouldn't really be possible. And I'll of course thank the NSF for funding as well. And if there's time for questions, I'd be happy to take them now.
Okay, thank you very much, Shweta. This was a very nice talk. So we do have one question. Okay. I'm going to read it, and then if there is time, I have one, if I have time to ask it. So the question is from Lorenzo Righetto, and he asks, do you expect results on modularity effects to change much in temporal network framework, and would your data support such a formulation?
Yeah, that's a great question, Lorenzo. So at the moment, at least, we're still quite limited in terms of the number of dynamic network data sets that are out there at this scale, because really to be able to do such a study, you would want to have a large number of data sets. I'm hoping that in the future we have more data sets, but we were able to look at that question for at least a handful, or I want to say maybe it was just a couple of data sets that are out there. And so yes, absolutely, we think in a temporal context, right, sort of the rewiring of edges can compensate for this, but we still do find that you need a fairly extreme level of modularity for that effect to be there. And I would say if any effects that are seen in other studies, the effect may not actually be because of modularity, but maybe because of other structures that are also changing at the same time, as the dynamics of the network are changing. But yes, indeed, it's a good question that will need continued work. So thank you.
So yeah, I think there is 1 more minute, so I can ask my question.
Okay,
so I was wondering, so in the first part of your talk, you mentioned this, like you said that what you were observing was due to the weak ties within your network, and you mentioned weak ties as having low weight. And it reminded me of the famous work of Granovetter, right?
That's right.
So I guess you have a little bit of a different definition of a weak tie. So you are talking about like low weight, which I suspect is a low frequency of interaction.
Yeah, low frequency, low duration. Yeah.
Yeah, low duration. Yeah. But still, I think can you make the analogy between the two? Because I mean, of course, he describes. Well, of course, I'm not if it's of course, but he describes weak ties as ties between people that typically belong to different groups, meaning these different groups that you also observe in terms of behavior within the same system, right? Yeah. Of individuals who have like different types of contact. And I mean, and can you make the analogy? Because in the last part of your talk, you did mention this part in which like you have people who have well, people, individuals who have different types of contact, right? And how this is important depending on the type of pathogen. So I was wondering, is there a relationship between the two or not?
That's a great question, Marta. I'm not sure yet, I think is the quick answer, because I do think people define weak ties in very different ways. Yes, you're right that the literature often defines them as what I would maybe call between module or between community ties are thought of as weak ties, those bridge connections, and other times it's between different types of, contact, as in the last part. What was interesting about our result is that that was the case no matter what the structure of the network, right? So, for example, a network didn't need to be highly modular, and it didn't need to have a lot of heterogeneity for the weak ties to really dictate what the structure looked like. So I'm not quite, I wouldn't say that they are the same, but yes, it's something where it's one of those things that really kind of turned our thinking on its head, because I would have expected there to be huge differences and variation between these social systems, and not to just be driven by the small contacts, right? Like we go to the grocery store and have those casual contacts. I wouldn't expect that those casual contacts to completely drive what my social system looks like.
Exactly,
right?
Exactly.
But yeah, but I think, yeah, more thinking to be done there for sure.
Okay, thank you so much.
Thank you.
So we are moving to our next speaker. So you, Fabrizio.
Yeah, do you hear me?
We do.
Okay,
you can share your screen.
Yes, just give me a second. Right, which one is? I think it's this one. Yeah. All right. See if you.
We can see your screen. Yes.
Do you see it?
Yes.
All right.
Yes.
Okay.
So our next speaker is going to be Fabrizio Lombardi from IST Austria, and he's going to talk about bursting dynamics and criticality in the sleep-wake cycle. So Fabrizio, whenever you're ready.
All right. Thank you. And I would like to thank the organizers for putting together such an amazing conference. I think that's a great work, and it's a lot of work. All right, so today I will show you some recent evidences suggesting that sleep regulation may depend on some tuning at criticality of underlying neuronal populations, which may explain some key features of sleep that do not fit into the homeostatic framework of sleep regulation. Okay, just to give you a brief introduction about sleep regulation. Sorry, does this work? All right. Okay, so according to the homeostatic picture of sleep and wake, so the alternation of these two different behavioral states is regulated by two processes. A circadian process C, which basically controls the alternation of periods with high sleep propensity and periods with low sleep propensity. And the other process is the homeostatic process, which is actually controlling the rise and dissipation of sleep propensities. At the neuronal level, there is a very simple model, the flip-flop switch model, that tells us how this alternation between sleep and wake happens. And this is simply happening due to the mutual inhibition of the arousal and the sleep promoting pathways. All right, those models are very simple, also very successful, but unfortunately, sleep phenomenology is way more complex, because during the sleep period, you have many transitions, irregular transition, among different types of sleep. For instance, you have transition between non-REM and REM. But most importantly, and I think not many of you knows that probably, is that you have many transitions to the awake state. So during sleep, you have many brief periods of awakenings, basically, right? So now, the question that still remains without an answer is why we do have so many irregular transitions, and especially why we should have transition to the awake state during sleep. So a potential explanation to this behavior came about 20 years ago when a bunch of physicists made a simple observation. And the observation is the following. So if you look at the duration of wake bouts during sleep, you realize that these durations are distributed according to a power law with an exponent that is about 2.3, 2.4, right? While if you look at the duration of sleep bouts, they are distributed according to an exponential-like distribution. Now, interestingly, if you take the brief awakenings and you consider them as the active phase of a dynamical process, and you take the sleep bouts and you consider them as the quiescent phase, you can immediately recognize that this duality of power law and exponential is closely reminiscent of self-organized criticality. In self-organized criticality, you have that avalanches have size and durations which are distributed according to a power law, but when you measure the distribution of quiet times between consecutive avalanches, you see an exponential distribution, right? So interestingly, this duality of power law and exponential distribution has been observed in many different mammalian species, and lately has been also observed in zebrafish. So what I'm trying to do here today is to show you some evidences supporting the hypothesis that these brief awakenings and arousals during sleep could be a projection at the behavioral level of some underlying tuning at criticality, which may provide and preserve the necessary flexibility to switch between different behavioral states. So in order to do that, what we do is following the brain activity in freely behaving rats for about 2 days, right? So we take the EEG that we recorded for 2 days across the sleep-wake cycle, and we try to extract signature of criticality from the EEG, basically, right? So we take the EEG, we divide the signal in windows of length W. Then in each window, we estimate the spectral power. And what we see is that the spectral power is basically dominated by 2 frequency bands. So the first one is related to the delta waves, and the other one is related to the theta waves. Now, the delta waves are related to sleep. So they will be our quiet state, basically, while the theta waves are related to wake and arousals, and they will be our active states, right? So now, in order to characterize the alternation between active and quiescent states during sleep, what we do is we consider the ratio between the power in the theta band and the power in the delta band, right? So we take this ratio, and actually what you see here, that when you consider this ratio, what you do is basically the temporal evolution of the EEG activity becomes basically you map that into a random walk. So what we do is the following then. So we consider bursts of we consider active states, those periods where the ratio is larger than a threshold H. Conversely, we say that we are in a quiet state when this ratio is smaller than the threshold H. And we simply give a duration to each of these states by taking the number of consecutive windows W with the same dominant wave times W. All right, so now we fix W and H, and we are ready to look at the duration distribution. So as you see here, when we look at the duration of active states, we see that they are distributed according to a power law. and with an exponent that is very close to the one that we measured at the behavioral level. On the contrary, the quiet states, they follow an exponential-like distribution, exactly as we see at the behavioral level. Now, importantly, those distributions are independent of the two parameters I was talking about before, W and H. So let's see when we take different values of H. So we take different values of H, and for the active states, we see that the power law behavior is independent of that parameter. The only thing that the parameter is controlling is the exponential cutoff of the distribution. And indeed, if you appropriately rescale the duration, you find a beautiful data collapse and the universal scaling function, right? The same thing can be done for the quiet states. You see that the distribution depends on the threshold H that you set, but actually, when you rescale the distribution but the average duration of the quiet state, they all collapse onto the same single scaling function, right? The same thing, same trick can be done when you look at different window size, and you also find a beautiful data collapse. So now I think this is pretty good and is pretty consistent with what we find at the behavioral level. So to finish, I would like to show you some nice correspondence between the dynamics of the active states and the earthquakes. So let's only consider the active states, right? We take the active states and we consider the threshold on the active state duration, right? This threshold D0. Then what we do, we decimate the events according to this threshold. So we take the threshold and we only consider events with the duration longer than that threshold, right? Then we measure the distribution of quiet times between those events. Of course, this distribution will depend on the threshold D0, right? This is what you see. And the same happens when you do this when you do this analysis for earthquakes, right? What we know from for the earthquakes is that this distribution only depends on the average rate, right? And indeed, when you rescale by this average rate, all the distribution, they collapse into the same scaling function, right? If we do that for our data, we also find a beautiful data collapse, and the universal scaling function here is a gamma distribution, which is the same scaling function that we have for the distribution of quiet times between consecutive earthquakes. Now, I would like to notice that since the scaling function is not an exponential, so already this is an evidence that consecutive theta bursts are significantly correlated. So what we find is that they are not only correlated, but actually that we have long-range correlation between consecutive theta bursts. Indeed, when we look at the fluctuation function, we see that the fluctuation function is scaling as a power law with an exponent clearly larger than 0.5. That means that there are persistent positive long-range correlation in the process. Okay, so I finish with this, and in summary, what we found is that if we look at the active and quiet state in brain activity, we find a very close correspondence with what we have at the behavioral level. That is, we have a duality of power law and exponential that is reminiscent of self-organized criticality. We also find another signature of criticality, which is long-range correlations, and together these two evidences, they clearly point to a role for brain criticality in sleep regulation, and in particular in the emergence of brief awakenings during sleep. Thank you. And I would also like to thank my collaborators at Boston University, Harvard Medical School, and the University of Malaga.
Okay, thank you very much, Fabrizio, for being on
Welcome.
time. So we have very short time for questions. I mean, nobody wrote anything in the chat, so I'm going to go ahead and ask a question that I have. And you didn't talk about this, but I mean, I was wondering. So you show this, after your analysis, your conclusion is like, well, I see these bursts, these correlated events, right? These long range correlations in events in the sleep-wake cycle.
Yeah.
So is this related? I mean, if people who don't sleep well, do they have a different behavior? Can you detect anomalies in the sleep cycle by looking at this? Like, are these long correlations lost? Or I mean,
right.
I was
okay. So we should distinguish what has been done at the behavioral level. The behavioral level is the sleep analysis that a sleep physician do, basically. So they see you. They put. So they look at your. So they basically classify your how much non-REM sleep you have, how much REM sleep, and if you wake up more than usual during the night, right? And so we know. So I know, but since long time that of course the correlation are influenced by, for instance, by sleep apnea, or if you have insomnia or other things. So of course those are influenced, but what has not been done is the analysis of such correlation in the way we have done, right? Okay. This is very new, and we haven't tried to do that on people, or like we try to do that on rats with where we lesion some area that is supposed to control like sleep, for instance, right? And actually we could find some differences in the exponent of the power law of the active state, but we could not find a significant difference in the correlation in this long range behavior. Right. But this is very much preliminary, and so I cannot tell more than that. Okay. But I suppose it does change. I mean, like, for sure. If you look at people with severe insomnia, they clearly show a different.
Yeah, of course. Yeah. Yeah, no, I was just wondering if it's something, a property of healthy people, if infants have a different behavior. I don't know. Yeah, no, it was very interesting. Yeah, thank you very much, Fabrizio.
Thank you.
So we are moving to our next speaker. So Emily, I think it's Emily.
Yes.
Yes, you're here. Okay.
My camera is not. Yeah.
It's not working. It's all right, as long as you can share your screen. Well, it's a PT, but I mean, we'll make the. Okay, perfect. We can see your screen. Okay, so our next speaker is Emily Skog. She comes from Universidad Pontificia Católica de Chile, or Pontificia Universidad Católica de Chile, sorry. And she's going to talk about modeling of cell aggregation dynamics during Killifish embryogenesis. So please, Emily, whenever you're ready.
Good afternoon. My name is Emily Skog, and I'm an undergraduate student for the Institute for Biological Medical Engineering, as they said before. And today I'm going to present in the in-silico modeling of cell aggregation dynamics during the anal Killifish embryogenesis. There are two types of bony fish, the non-anal and anal. Each have very different developmental processes. The non-anal has a high-speed embryonic development. The epiboly occurs at the same time than the gastrulation and the axis formation. We can observe in the video, we cannot discern between each process. In comparison, the anal fish, in this case the killifish, which is the one we're studying, has a much slower development due to its adaptations to seasonal droughts. Fortunately for us, this allows us to study this complex biological system as each embryonic process occurs separately. The stem cells, which are the green dots, initially are dispersed all around the egg, to then aggregate in a single cluster at the pole of the egg. Then the axis forms and eventually the proper embryo. How this aggregation process occurs is what is currently unknown. So our main question is, can modeling predict the minimal parameters necessary for embryonic development? To address this question, we have developed a biophysical model to study the aggregation dynamics and to have a better understanding of the killifish embryogenesis, but also in other vertebrates. The stem cells initially move in the epithelial enveloping layer and the yolk. They move tangentially between these layers. Interaction with them is the same as between the stem cells. We base our mathematical model in Smith's 2016 paper, where there stated 4 parameters that control this aggregation process: the intrinsic motor force, which is the force that propels the cells forward; cell adhesion, which is the main parameter for the aggregation process. This is the tendency of cells to stick together or to surfaces. And it is conditioned by the cell-cell adhesion energy and the cell substrate adhesion energy. And it works as a sort of spring. When the distance between the cells is twice the radius or more, they tend to attract to one another. And when the distance is less than this, they tend to repulse. Another parameter studying is a contact inhibition of locomotion, CIL. This is a repulsive interaction where the cells drive away from each other when they come into contact at a certain repolarization angle. The repolarization rate is conditioned by this angle, a polarization force, a diffusion rate coefficient, and a Gaussian white noise. And the last parameter we're studying is the cell density over the egg sphere. We conditioned this during the simulations, changing the embryo's radius, but maintaining the number of cells constant throughout. So we then ran a series of numerical simulations scanning all these different parameters. And we can see that the cell density is 0.2, close to the experimental value, and at a higher cell adhesion, clustering does increase, but we do not form a single cluster. But at a higher cell density and a higher cell adhesion, we do form a single aggregate. In this case, a stringy one. Analyzing the rest of the parameters, for both densities, we can visualize similar trends. As cell adhesion increases, the number of clusters decreases, and as the contact inhibition of locomotion increases, the number of clusters decreases as well. Meaning that there is a relationship between the cell adhesion, the contact inhibition of locomotion, and the density. However, we can only visualize a single cluster at a higher density. So then, we carried out a series of other simulations for a longer timescale. And we plotted the number of clusters over time, and analyzing this log-log plot, while the cell is clustering, the linear fit follows the power law. But when the number of clusters decreases to a few, we lose this linear fit and form another power law. And then looking at this video for the density 0.2, which is close to the experimental value, in this case, we do form a single aggregate. And different from the past simulations, the morphological shape of the aggregate is similar to the experimental one. However, in the in-silico simulations, we could see that small clusters fuse into larger clusters to then form the single cluster. But in the in vivo aggregation process, we can see in this video that single cells coalescence into the large cluster, meaning that our simulations still do differ from the in vivo process. So we predict that there are other intrinsic factors or external cues in play during the aggregation process, such as chemotaxis, which is a migration of cells due to a chemical signal, durotaxis, which is a cell migration due to a rigidity gradient, and lateral inhibition, which is a mechanism of cell inhibition on an adjacent cell's activity. We're currently exploring all three mechanisms. So we can conclude that the anal killifish thus provides a unique opportunity to study these developmental processes. As I mentioned before, each embryonic process occurs separately, making it easier for us to study them. Our computational model has shown to be a powerful tool to model this anal killifish embryogenesis. And thanks to this, we can demonstrate that the self-organization of embryonic cells into a single aggregate can spontaneously occur with a simple combination of physical parameters. In this case, cell density, cell adhesion, and contact inhibition of locomotion. But further implementations will be directed so that we can better recapitulate this embryonic process, including the biological parameters such as chemotaxis, durotaxis, and lateral inhibition. I want to give special thanks to the collaborators in this project and main tutors. This investigation was funded by the Chilean Ministry of Science. Thank you very much for listening, and I'm open to any questions.
Thank you very much, Emily. So I don't see any questions in the chat. I don't know if anybody in the audience wants to turn on the microphone. If not, I will go ahead and ask a question. Yeah? Okay, so I was wondering, Emily, so you at the beginning of your talk, right, you showed these two different annual and non-annual fish with two different ways of developing. Like in the first one, you cannot see like an axis or a direction or like something that breaks the symmetry, and in the other sort of you can. So I was wondering, with your type of modeling, would you be able to model both processes by changing some of the parameters or not? I mean, because do you understand what I'm saying?
There's my camera.
Oh, your camera is working.
Yeah. Sorry, can you repeat the question?
Yeah, I was saying that you use it to model the embryogenesis of this killifish, right? Mm-hmm. The one in which, like during the developmental process, right, you see this formation of this one cluster, right? In the other case, I think you said like it was non-annual, or I don't remember. Like the other case that the case that you showed at the beginning. At the beginning, you don't see anything that breaks the symmetry, right? You don't see like a polarization or anything in the same way that you see it. But would your model be able to reproduce that? What would it take for your model to be able to reproduce that? Like, that's what I'm asking. Like, what would you have to change?
That's the idea, because it occurs concomitantly. We cannot, because what we're doing now is like analyzing the in vivo videos and everything, and we cannot visualize what is occurring with all the stem cells and the movement of cells and the axis formation. We cannot discern between each process from what we can see now, and with the anal fish, we can look at each process. Okay. I don't know if I can explain this. Like no. You said you can't. Video, you can't because you cannot make a comparison. You don't know, basically. No. You wouldn't be able to know. So okay. I thought that it was more a matter that because you don't see a polarization, you cannot attempt to model that polarization, rather than you're not being able to measure. Oh, okay, I see. Okay, perfect. Mm-hmm. Thank you very much, Emily. So thank you for being on time. Also, we have saved a little bit of time, so now we are on time instead of being a little bit late. So this is so we move to the next speaker. Thank you very much, Emily. So our last speaker for the session is going to be Ian, I believe.
Yes.
Yeah?
Yes, me.
Okay. You're here. Okay.
I am.
Okay, perfect. So, so our last speaker for this session is going to be Ian Leifer. You have not put your origin, but if I don't know, are you from City University in New York?
Yeah.
Okay.
Yeah.
Okay, perfect. So he's coming from New York City University, and he's going to talk about vibration symmetries to uncover the building blocks of biological networks. Okay, please.
Yeah.
Whenever you're ready.
Thank you. Thank you for the introduction. And yes, hello everyone. My name is Ian Leifer, and I'm a graduate student here in City College of New York. My advisor is Hernan Moxie. And here I'm going to be presenting our project, how we apply vibration symmetries to biological networks to uncover building blocks and study how some of them function. So it's really important it's a really good approach to studying networks by decomposing them into building blocks, because it really makes sense. You take the big network that has like very complex behavior, and you break it into small parts. You say, okay, so this small part functions like this, this other one functions like that. Then you study how they interact, and then you have a good idea of how the whole system functions. So it's a really good way to approach this. There have been few approaches to this so far. There is modularity. Another very popular approach is network motifs. But so far, there was no approach that is based really on function, on like what biologically does this building block do. So here we offer the approach that is based on symmetry considerations, and using that we apply it to some real networks, real large-scale networks which haven't been done before. We find some building blocks, we classify them. And I also want to preface what I'm going to say by saying that here and just in our papers, we applied it to transcriptional regulatory networks, that is gene networks. And the examples that I'm going to use are from bacteria, but really we also applied it to neural, metabolic, all kind of biological and not even biological networks. And it can be applied to basically to any directed network. So I'm going to start with just a small introduction to the ideas that we use, because symmetry, the word symmetry is used for many different things. So let's consider this small circuit, which is the example from E. coli, just like small piece of transcriptional regulatory network from E. coli. Let's look at these three genes here. So if you look at if you look at these parts of the network, you wouldn't really see any symmetries. You look at these two genes, you can just switch them because there is a self-loop here, there is a directional link here. You can switch these two because there is a big part of the network that's going to be changed. You can switch these two, obviously, because there's no self-loop and etc. So seemingly there are no symmetries in here. So well, how do we go about this? Well, if you look at the information that is processed in the network, it turns out there is some symmetry. So if you look at this node, buyer, and if you look at the inputs, so you see there is a self loop and there is a link from the green node here from CPXR. So you will see that buyer receives from itself and from CPXR, and you see how it keeps receiving, and then the green link has the self loop here and it keeps going here. So what you will see if you look at the inputs of this node, you will see this thing which is called the input tree. So the input tree of this node looks like this. If you look at this node, it also receives from red and green here, and then you keep going and you would get the same tree as before. So this is really interesting because looking at this network, there are no symmetries in here, but now suddenly you actually look at the topologies of the input trees, and what you see is that some of them have the same topology, or if you want to be like graph theoretical, they're isomorphic. So if you look at the input trees of all the nodes in this network, you would find that, for instance, these two are isomorphic, these three and actually four, and are isomorphic, these two, not even two, but like the colors are have isomorphic input trees. So and this helps to decompose the network into into these parts. So these parts are called fibers. So what is so special about fibers? Well, it actually has been shown that fibers synchronize. That if you take the directed network, which each node is really, for instance, in this case, transcriptional regulatory network, each node is concentration of the mRNA of the gene production, but it can be neural activity, it can be like neuron that's spiking, it can be whatever it is. So each node is represented by the ODE, and if you look at the activity, then what you see is synchronization, and that's what we see actually here. We just put one simulation. For instance, we take CPXR, so this gene we put it on, and then we see that this dynamic actually stabilizes and goes together, and it keeps staying together. So this is robust, stable synchronization in this specific case, which is really cool. So this is like the core concept here. Now I'm going to just also briefly introduce one small thing because I'm going to need it a bit later. So the branching ratios. So if you look at the number of nodes at each layer in the input tree, you can find the branching ratio as a number of nodes in the layer I+1 over the number of nodes in the layer I for I going to infinity. So basically the number of nodes in the next layer to the nodes in the previous layer for the far away. So here, the simple case, always there is one node in each tree on each layer. Therefore, n is going to be equal to 1. N is the branching ratio. Here is going to be 1, 2, 3, 4, 5. Obviously, then you got I+1 over I for I go into infinity. Also, branching ratio is also 1. So this is one of the things that we're going to use to classify. So this is as far as methods go. Now, some of the things that we discovered, and I'm only going to cover part of what we discovered. And one of my lab mates is actually speaking in one of these days, and he's going to go more into detail about how building blocks interact. But I'm only going to cover the building blocks themselves. So this is there are two types of building blocks that we discovered here. So the first one has integer branching ratios. So we define a building block as the synchronized nodes. So we take a fiber and the inputs of that fiber. So in this case, the fiber was these two genes, and we take one node that is an input of them. So there is only one node that sends to them. So we get this topology of the building block, or we can get this topology of the building block, which is well known by Fan, which is one of network motifs, and then this extension of it, and etc. And then you can look at the input tree. In this case, it's finite. So we take the branching ratio as 0, and we use these fiber numbers here, and then n is branching ratio, L is number of regulators. So n is equal to 0 in this case, and all three of them. L is equal to here 1, 2, and 3, because different number of regulators. Now the second example, and I guess I'm just going to, I just give these examples because those are the ones that we found in E. coli, but there are much more if you look in the paper. So you get like two nodes here that have self loops, and they have back and forth connection. And if you look at the input tree here, you would get the branching, the doubling on every layer. So because you get this doubling on every layer, the branching ratio is going to be equal to 2, sorry, and number of regulators is equal to 1 in this specific case here. So this is classified by 2, 1. Obviously, there is more generalized topology. So this is really cool because we can actually find really big building blocks. Like so far, there is like a limit. If you take network motifs, there is a limit on smaller sizes, and then it's hard to find them. Here, you can find any sizes, and this is all based on function. Now, the second type of building blocks that we found is non-integer branching ratios. So in this case, we have to include if any node in the fiber belongs to a strongly connected component, that is, if there is like a longer loop, we have to include the whole loop too. So I'll speak specifically about this example in the interest of time. So this would be the building block in the definition before, but now there is this extra loop that we want to add. So now if we add this loop, we actually get this input tree that is a little trickier. So we get after certain delay, input tree kind of repeats itself on itself. And if you write down the number of nodes on each layer, you will see that it actually follows this generalized Fibonacci sequence of the delay. So what you get is this generalized Fibonacci sequences, and this branching ratios, you get Fibonacci numbers. And this is also true for like delay of length d, and this is another case here. So now I'm going to talk about function. So I want to discuss how these building blocks function, and I'm going to just talk about two basic ones, in the interest of time. So one of the basic ones. And this is not really the building block. This is the core part of the building block, and then everything else kind of comes from here. Again, just to be brief. So let's take one node with a self-repression loop. So repression case is very clear what it is in case of transcriptional networks, and this is what we study here. So we take this, then the input tree is going to be just chain. So it's 1, 0. And the analogy, so what we want to do is in biology, and specifically with genetic networks, what they often do is they study analogy of electric circuits to look at like Boolean models to simply look at the simplified dynamics. And that's what we did in one of our papers. So we consider this not gate that just connects to itself. So there is like a state of this not gate, which yeah, and then it goes, goes back in. So in the Boolean model, it's just obvious what it is. So state at moment t is equal to state of at moment t minus 1, but not state of at moment t minus 1. So if we start at 1, then we go into 0, then we go in 1, 0, 1, or like this. So we get this oscillation here. Now, if you look at the continuous model, which is really all this behind this genetic network, there is a system of ODEs. And if you look at the continuous model, you will also see oscillations here too. So this actually is the evidence, which is very brief, that these type of circuits with self loops with delays can function as clock. So this one type of circuit, and the other type of circuits I'm going to cover is so there are two nodes that repress each other, and they have external inputs, and this is analog to a SR flip-flop. This is a toggle switch. So this is the smallest memory unit that goes from 0, 1 to 1, 0, meaning state 0, 1, 1, 0, and changing by giving different set and reset, you actually can jump from one state to the other and back. So this is the simplest memory unit. So basically what we found is that there are clock circuits and there are memory circuits, and this can actually help to decode the full network. And I'm going to just wrap up here. So conclusions are this. So first, vibration symmetry, the stuff I talked about in the beginning, is a type of symmetry that actually can help to analyze biological or any directed network. Number two is we can uncover building blocks that are related to functional building blocks that are related to synchronization based on these symmetries. And the last thing I want to say is that this approach is not only theoretically principled, but it's also very heavily algorithmically supported, because these things have been researched in computer science for a long time, and there are many different approaches to how to do this. So it's very heavily computationally, it's very easy. So for further reading, I want to refer you to our two recent papers, one on PNES and one on PLOS, and algorithm is available at 2 GitHub links here. So thank you for your attention, and I'm happy to take any questions.
Thank you very much, Ian. And thank you for being exactly on time. So I don't see any questions in the chat. So I'm very curious. I could be asking you many questions about this. I couldn't quite follow some of the explanation. So my question is very basic. So by depicting the network like this, right? What are you able to do that you wouldn't be able to do otherwise? I'm sorry. I mean, this is why I'm saying it's very basic, because it wasn't clear to me. I know you can do many things, but I mean, I see that representing the network in another way, I could do exactly the same. So I'm just wondering what it is for you, like what, yeah.
Right. No, I think this is a great question, and yeah, thank you for asking. It's I think there are two things that are really important here. So one is just the representation itself and why is it good. So input tree, she tells you what kind of information node has about the network. What does it know about the network? What does it not know about the network? So this is just why is it good. But really, like what's the good thing? What's the use of this? So the use of this really is that you can actually take the network, you can decompose it into these parts, and actually simplify. Like I didn't put, I have some slides on that, but I can't really talk about all of this. But you can, like for instance, we took network of E. coli, we basically broke it into small parts, we decomposed it, and then we almost we were very close to really giving an explanation of how the whole thing functions, which is like a 1,000-node network, which no one did before. So I think this is really convenient because it can actually in terms of analysis, I think it's a really convenient tool. Yeah. Did that make sense?
No, it does. It's a beginning. It's a beginning. I would have more questions.
No, sure.
Well, but we are sort of running out of time, and I don't know if there are more sessions after this, and I don't want it to debate. I think it's very interesting. I thank you for the references. I think it will be a nice read. So I would like just to thank all of the speakers in the session. I think it was a very nice session. We saw very different things, very different complex system things, and a lot of food for thought, I think. And I thank everybody for participating and coming and listening as well. And it's unfortunate that we cannot clap. I clap for all of you because I think you did a great job. And well, maybe we see each other in one of these gathering rooms. or in another session. And that's it from my side.